Okay, let's continue. Sorry, I ran out of time on that last video. So then we have our DNA organized in chromosomes, and we start with the double helix at the top. And sorry about the noises. This is somebody else's slide. Then you have the DNA is wrapped around the histone proteins, which are the yellow things. And that is simply like threads on a spool, followed by the chromatin. And a chromatin is organized into a long, thin fiber. And then what will happen is it will actually condense further during mitosis. And so at the end, you have a double stranded chromosome with duplicated mitotic chromosome. So here is another view of it. So after DNA duplication, chromatin condenses, and basically what that is the coiling and folding in order to make it into a smaller package. So DNA, chromatin, and then the mitotic chromosome. And here you can see it in a view as well. It is actually during metaphase when you have the most visible and condensed chromosomes. And this is somewhat what they look like under electron. Well, this is what they look like under the electron microscope. Quite amazing. What will happen in the overall process is we will start with a chromosome that is then duplicated, and then in mitosis you have the separation of what are called the sister chromatids. So you have in the cytoplasm of prophase, a mitotic spindle will form, and what's happening is microtubules are forming between the centrosomes. And I want you to notice again, here's the aster. That prefix is star. And so sometimes it looks like a star because of its rays. And then centrosomes are moving apart. And the way that they are moving apart, the mechanism by which they move apart is because the lengthening of microtubules occurs. And we will look at that in the next a little bit later. I'll show you. Okay, so we have prometaphase, and in prometaphase, prometaphase, you've never heard that before. Well, guess what? New phase. We always did prophase, metaphase, anaphase, telophase in regular bio, but actually in our AP, we add a phase called prometaphase. And in this, the nuclear envelope disappears. And then you will start to see the microtubules are actually interacting with the chromosomes, and you see that they can actually attach. So, what you have are spindle fibers that are made of microtubules. And what will happen is at this point, you'll have the centrosomes are completely at opposite poles. And the spindle fibers extend out from the poles to what we call the equator, which is the middle of the cell. There are things called kinetochore microtubules or kinetochore fibers that actually attach to a chromosome, attach to the centromere. And then we also have non-kinetochore microtubules that do not attach, and you can see those off here on the side. No connection to the chromosomes. So you have a structure actually on the chromosome called a kinetochore. And that's where that comes from. And you can see this back in this image right here. The kinetochore is the structure to which the kinetochore microtubules can attach. So the middle here is the centromere. These are kinetochores. These are kinetochore fibers coming off. And then these are sister chromatids. Now, this whole thing is one chromosome. If you have a centromere, you have a chromosome. And that is something that you have to start getting into your head, because later on we will actually talk about numbers of chromosomes, numbers of centromeres. And if you don't have that knowledge that a centrosome means, or a centromere, sorry, means a chromosome, then you will become lost easily. I love these slides. This one is just amazing to me. You can see the microtubules and the chromosomes. They look kind of like cheese curl puff things. And then you can see right here that the non-kinetochore microtubules will overlap, and you will see the microtubules attaching